Hello, I'm here to briefly present our work entitled Brain-derived neurotrophic factor serum levels following ketamine and esketamine for treatment-resistant depression. My name is Gustavo Leal. I'm a psychiatrist at University Hospital at Bahia, Brazil, and together with my colleagues from Bahia and São Paulo, we've developed this work that I'm presenting now. Just some quick information for background. And ketamine is the racemic mixture of the isomers S-ketamine, usually called esketamine, and R-ketamine. Both the racemic mixture ketamine and the esketamine have been shown to have a very fast and potent antidepressant action. And there is also data showing that ketamine might influence brain-derived neurotrophic factor, BDNF, and this effect might be involved in the antidepressant action of ketamine. BDNF is a growth factor from the family of neurotrophins, and it's involved in many processes involving the central nervous system and peripheral nervous system, including development, differentiation, and survival of neurons, and also in neuroplasticity. Uh, It has been shown that in depression, BDNF levels might be reduced in some brain regions and in peripheral blood, and that its levels might rise together with the improvement of depression. And some animal studies suggest that not only the BDNF levels are associated with the improvement of depression, but that it might be necessary for the effects of these drugs, including ketamine. So, for this reason, BDNF might work as a biomarker to help guide antidepressant treatment. So, in this study, we aim to assess the impact of ketamine and esketamine on BDNF serum levels and to analyze if there is a differential impact of ketamine and esketamine that it might even also to help to understand the mechanism of action of these drugs. Also to analyze if the BDNF levels associated with the antidepressant effect in patients with treatment-resistant depression, and also if the levels of BDNF might work as a predictor of response. Uh, this study is derived from another trial, a non-inferiority trial, comparing the efficacy of ketamine and esketamine for treatment-resistant depression. In this study, the main inclusion criteria were age 18 or older and a diagnosis of major depressive disorder, MDD, and being considered as treatment-resistant depression, in this case, as having one failure or more of treatment of antidepressant treatments. The main exclusion criteria were psychotic disorders, cognitive impairment, unstable medical illness, and substance use disorder. The Montgomery-Åsberg Depression Rating Scale, MADRS, was used for assessing depression severity, and BDNF was measured in serum. The statistical analysis, we used a general linear model of repeated measures for assessing MADRS and BDNF levels along different time periods and to compare the impact of the different intervention groups. And we also use Spearman correlation for some other analysis. Here we can see the design of the study. So patients were at baseline or right before the infusion were assessed with MADRS and blood was collected for analysis of BDNF levels. The intervention consisted in either a 40-minute IV infusion of ketamine 0.5 milligrams kilo or esketamine 0.25 milligrams kilo. Then they were reassessed 24 hours later for moderate BDNF, after 72 hours only for MADRS, and after 7 days for MADRS and BDNF. Here we can see our results. On the left, the flow chart of the study. We have 79 patients assessed for eligibility, and 63 were randomized and allocated for either esketamine or ketamine. As we had some blood samples that were not fit for analysis, we had A total of 26 patients in the esketamine group analyzed for BDNF and 27 in the ketamine group. Here in the table of clinical characters and demographic characteristics, we can see that those characteristics were well balanced between the two groups as for age, gender, number of depressive episodes, and number of treatment failures, as well as for baseline MADRS score. There was really similar between the two groups. Here we can see our main results, and for using our model, we found that there was a significant change in MADRS score over time, and in visualizing the graphic, we can see that this change was a decrease in MADRS score, so improvement in depression, mainly from baseline to 24 hours, but this improvement was maintained after 3 and 7 days, and that our model showed that this change was not different between both intervention groups. So both ketamine and esketamine has a similar antidepressant action in this sample. But regarding BDNF levels, we see that there was no significant change in BDNF over time, and this also there was not a difference between the two intervention groups, total and a long time, since there was no interaction between those factors, time and intervention group. It's very clear when you see the graphics, where you can see the mean BDNF levels, and we can see that the evolution on time was almost a horizontal line, being very clear that there was no change in BDNF. Some other results, we found that the BDNF levels at baseline did not predict change in MADRS scores, and also BDNF levels in other time points could not predict. We found no correlations between MADRS scores and BDNF levels at any of the time points. So, and also that BDNF levels do not correlate with age, and we did not find difference between males and females regarding BDNF levels. So we consider it was an important study since it's the first human study directly comparing ketamine and esketamine regarding impact on BDNF levels in TRD patients. And summarizing our findings, we can say that the two drugs did not impact serum BDNF levels, and that there was no association between BDNF levels and depression severity. Comparing with other studies is hard because data vary a lot between the studies, but mainly one study that found a significant impact of ketamine in BDNF levels found that the measurement was 4 hours after the intervention. It was a study conducted by Haley in 2014. So in our study, we didn't measure that early, so we cannot rule out that these changes might have happened before the 24 hours. But also, many other studies did not find any association between treatment with ketamine and changes in BDNF. So that's a field still open to investigation. And some questions are important when studying BDNF. If those measuring at plasma would be better than at serum, each mean has its advantages and disadvantages. Plasma might be better for detecting small changes, but also more unstable for this analysis. And also, I think one of the main questions if the peripheral levels of BDNF also really reflect what's happening in the central nervous system. That's a question we still don't know the answer, and it's fundamental to understand if these measurements in peripheral blood are useful for what we want to detect. We have some limitations in our study. The mains was not designed for this outcome. As I said, it's derived from a study designed for analyzing efficacy of ketamine and esketamine. Also, we didn't have a placebo group that would have to analyze these findings. We did not standardize the periods of fasting, since food can impact BDNF level that might have been important. But since the measurements were made at the same time of day, we don't think that did impact. And also we did not standardize concomitant medications, since we in our study we reflect a natural world scenario. So this was it. Um, you can see more in the complete study that was published and last year. And I thank you all for the attention. I'll be available for answering questions at the scheduled meeting. Thank you.